Hello everyone, welcome back to Med for You channel. Today, we're diving into the fascinating world of genetics and exploring the leucine-rich repeat kinase 2 or LRRK2 for short. Let's get started. LRRK2 is a gene that produces the tartarine protein active in the brain and expressed in multiple tissues involved in both inflammatory responses and defense against infectious organisms. Further, studies shows that the RRK2 gene mutations are the most common cause of autosomally inherited Parkinson's disease. Parkinson's disease is the second most common neurodegenerative disorder, and at least 4% of familial PD cases in specific populations like North African Berber and Ashkenazi Jewish patients are due to LRK2 mutations. Compellingly, LRRK2 mutation may play a role in natural selection since people with mutated LRRK2 protein have a greater chance of acquiring Parkinson's disease than those who do not. And I bet you are wondering how LRRK2 evolved. Well, to answer that, LRRK2 belongs to the ROCO gene family, which produces large proteins with multiple protein domains. In vertebrates, there are four ROCO genes: the LRRK1, LRRK2, MFHAS1, and DAPK1. Phylogenetic analysis have revealed that LRRK1 is the closest relative of LRRK2. This relationship is supported by their structural similarities as both genes encode a TKL group kinase domain, which is absent in the other two vertebrate ROCO genes. And what is more interesting is that according to the phylogenetic analysis, it was discovered that LRRK2 originated from a duplication event that took place after the separation of protostomes and deuterostomes. The genes LRRK1 and LRRK2 are present in all deuterostomes. However, in protostomes, only LRRK1 is present. Therefore, LRRK2 is a gene specific to deuterostomes and arising through gene duplication after the protostome-deuterostome split.
Now let's discuss the mechanisms behind this genetic mutation. First, the involvement of LRRK2 with Parkinson's disease (PD) is significant as genetic variations in LRRK2 influence PD susceptibility and its interactions with other PD-associated proteins. Understanding the mechanisms behind LRRK2 in PD pathology is crucial for developing effective treatments. LRRK2 mutations can also cause mtDNA damage in neural cells even before PD symptoms appear. The correction of the genetic mutation in identical cells eliminates the mitochondrial phenotype and provides a compelling evidence of LRRK2's role in causing mtDNA damage. Moreover, LRRK2 is involved in pathways affected in PD such as autophagy, mitochondrial dysfunction, and immune system response. Genetic variations affecting LRRK2 function can lead to abnormal activity in these pathways, contributing to PD development. Additionally, individuals with PD carrying the LRRK2 G2019S mutation exhibit a higher frequency of non-motor symptoms, including olfactory dysfunction, anxiety, and depression, compared to idiopathic PD. These non-motor symptoms could provide insights into PD's underlying pathology and guide the development of new treatments. Moving on to the relevance of LRRK2 mutations in the field of molecular medicine. Recently, researchers have found similarities between inflammation occurring in both Parkinson's and Crohn's disease patients. Their LRRK2 gene expression was high on the peripheral tissues and the brain, so mutations in LRRK2 were also found in CT patients. Further research has also unraveled LRRK2 gene variants, two of which are missense alleles. Scientists are also close to modeling the pathophysiology of PD with greater accuracy. We owe all of this development to our better understanding of the mechanisms of the emergence of LRRK2 gene variants. And from here, the future is bright for developing treatments involving disease modification via LRRK2 gene inhibition. And that's everything you need to know about the gene of the day, LRRK2. Thank you, and we hope that you feel wiser now. Always remember, stay curious.